Okay, so welcome back to this next video in which we are discussing endocannabinoids and the cardiovascular system. Okay, right, so we're currently in the process of discussing the role of endocannabinoids in the cardiovascular system. Now, we've seen how there seems to be this very different effect of endocannabinoids if they're produced locally versus whether they're produced systemically. Okay, now the other important thing that I have said is that there is no endocannabinoid tone. It does not seem as though the endocannabinoid system is important in normal cardiovascular physiology. It seems to be very important in pathophysiology, specifically in times of ischemia. Okay, so we've discussed that if a portion of tissue becomes ischemic, then potentially it will start producing endocannabinoids, and this will cause the vasodilatation of the local blood vessels that lead to that portion of tissue. Okay, so we've discussed about how it produces vasodilatation of isolated arteries. The assumption is that it will be the same for arterioles, the smaller blood vessels that lead to specific portions of tissue. Okay, and this then would lead to an increased blood flow to that portion of tissue. Okay, right, and hopefully, therefore, reverse the ischemia. We'll also see later on how the cannabinoid type 2 receptors seem to be very important in protecting you against ischemia reperfusion injury, and we'll come on to that later on. Okay, now, the other thing that I just want to do is extend this a little bit. So I want to talk about also what happens if you get ischemia in the heart. Okay, so let's now think about ischemia in this specific special organ, the heart, because here the crisis becomes a little bit more interesting. If the same thing happened in the heart and the heart cells were to start producing endocannabinoids in this time of ischemia, then what would the endocannabinoids do? Well, just like everywhere else, they would cause vasodilatation of the blood vessels that lead to that specific area. Okay, but also the endocannabinoids would act on CB1 receptors on the surface of the myocardial cells themselves, and what that leads to is negative inotropy. It leads to contractility going down. So basically it makes the heart muscle cells work less hard, okay, which means they're going to consume less oxygen. Okay, so you get a double effect basically. This is also going to help protect the cardiomyocytes. Okay, you've got ischemia. One, we're trying to increase blood flow, but also we want to decrease oxygen consumption, and one way of doing that would be to make the heart work less hard. So there's a specific example of how it might protect the heart during ischemia. Okay, now let's think about what would happen if it went up systemically. Okay, so basically if endocannabinoids rise systemically, then obviously a very different effect is going to happen. Okay, and a very different effect is necessary, because if you think about what would happen otherwise, if endocannabinoids went up systemically, what you would expect is blood vessels to start vasodilating all over the place. Okay, and that would probably lead to a terrible collapse of blood pressure. Okay, so there's real danger there if this system was abused and used systemically. Okay, so what seems to happen is the system has a way of combating that, which is that if it goes up systemically, then to stop this disaster, what happens is the endocannabinoids activate the central nervous system to activate sympathetic tone. Okay, so sympathetic tone goes up and reverses the effects that the endocannabinoids would have on each local area, basically. Okay, so one, sympathetic tone going up to the heart is going to cause the heart to beat harder and also to beat faster, so heart rate is going to go up. Okay, so sympathetic tone is going to go up, heart rate is going to go up, and also contractility is going to go up. So that will reverse the effects of the endocannabinoids on the cardiomyocytes, and in fact swing it completely the other way to the heart working harder, basically. In addition, in the blood vessels, it's going to cause contraction of the vascular smooth muscle cells rather than vasodilatation. So again, it overpowers the local effects of the endocannabinoids and completely swings it the other way. So contraction occurs, and this causes vasoconstriction, basically. Okay, so if you get systemic rises in endocannabinoids, there seems to be this way of protecting you against the disaster that would happen if you just had the local effects occurring everywhere, basically, okay, which will cause most likely shock, i.e. blood pressure thundering through the floor, okay, which is very, very dangerous. Okay, so those are the difference between the local and systemic effects of endocannabinoid production. Okay, so what I now want to talk about then is another experiment which talks about exactly this, which is the question of what happens if you inject anandamide systemically. Okay, so this has been done again in experimental animals, and I think it's again in rats and mice that this is done. Okay, so there is a big difference here between doing it in a conscious animal and doing it in an unconscious animal. So we'll start with doing it in a conscious animal. So basically, let's say now that we're going to give anandamide by an intravenous injection into the circulation. Okay, so intravenous injection. So, and we want to see what effect is this going to have on the blood pressure of the animal. Okay, well, let me just talk firstly about how blood pressure is monitored. How blood pressure is created. Okay, and how what influences blood pressure. So let me just give you a very basic model of how the cardiovascular system works. Okay, and it's going to be a very basic model. So here, this box is going to represent the heart, okay, and also the pulmonary circulation. Okay, so the heart, broadly speaking, is just going to take blood from the venous system here. Okay, so this is going to represent the venous system, and let me colour bits of this in. So here's the venous system in blue over here, and basically the heart is going to take bucket loads of blood and dump them from the venous system into the arterial system here. Okay, and each bucket's worth of blood will be a stroke volume's worth, so that's a key term here. So SV stands for stroke volume. So the heart is going to take a stroke volume's worth of blood from the venous side and dump it into the arterial side. So this is the arterial system here. And of course, in between doing that, it's going to pump it round the lungs, round the pulmonary circulation, and oxygenate it, and then it will pump it out into the arterial system. But to keep this nice and simple, we'll just omit the pulmonary circulation. Okay, right. So what then happens is when the heart pushes a stroke volume's worth of blood into the arterial system, the arterial system originally expands to take that blood. So the aorta is a fantastically elastic artery. It just bulges basically to take the blood on board that the heart is pushing into it. Okay. And then what's going to happen is gradually, whilst the heart is now relaxing, the aorta will elastically recoil, and it will gradually push the blood out of itself and through other blood vessels. Okay, so through smaller arteries and then through even smaller structures known as arterioles, and then the arterioles will eventually divide into capillaries, and these are where the actual gas exchange occurs with the peripheral tissues. So here are the capillaries, and then the capillaries will reconverge into venules, and then the venules will drain into small veins and then into the big veins again. Okay, so here's the venous system, here's the arterial system. We'll put the capillaries in turquoise in the middle, and then you have smaller blood vessels, which are the arterioles and the little arteries here. Okay, and then the venules, which are the vessels that drain into the venous system. Okay, so basically what I really want to talk about is these vessels here, okay, the little arteries and then the arterioles, okay, these together are what are known as the resistance vessels. These determine how much blood can actually flow from the arterial system through the tissues basically, okay, because as I say, when the aorta receives this stroke volume's worth of blood from the heart, it expands, okay, it bulges out elastically, okay, and then gradually it's recoiling under elastic recoil and gradually forcing the blood through these smaller blood vessels, which will then take the blood back to the venous system. Okay, now if these vessels all over the place are very, very constricted, then it will be much harder for the aorta to push blood through them, and it will be much slower process for the blood to be pushed out of the arterial system into back into the venous system eventually. Okay, so these blood vessels are very, very important in controlling how quickly blood can return from this bulged aorta back into the venous system. Okay, right, so now let's discuss what controls then blood pressure. So basically, there is a fantastic difference between the pressure in the arterial system and the pressure in the venous system. So central venous pressure, or CVP, is approximately 8 millimeters of mercury. Okay, so you've got very low pressure in the venous system, whereas the mean arterial pressure, MAP, is around 90 millimeters of mercury, and it has to be a mean arterial pressure because it's always changing basically. When blood has just been forced into the arterial system, of course the pressure will be much higher in the arterial system. Okay, so this is a mean pressure. Now, how is that pressure difference maintained? Well, of course it's maintained by the heart. Okay, the only reason this pressure difference exists at all is because the heart is always taking buckets of blood from the venous system and forcing them into the arterial system. Okay, if you just stopped the heart working, okay, if you took it out, then what would happen is blood would move from the arterial system to the venous system, and everything would just equilibrate. The pressure here would be the same as the pressure here, and flow would stop. Okay, so the only reason this pressure difference is maintained is because the heart is continuously moving blood from the venous system to the arterial system. Okay, so if we want an equation for mean arterial pressure, basically there is this very useful equation, which is that mean arterial pressure is equal to the cardiac output, and the cardiac output just means how much blood the heart moves from the venous system to the arterial system in a minute. Okay, so basically cardiac output is the stroke volume, which is the volume of blood moved from the venous system to the arterial system each beat, times how many beats you make in a minute. Okay, now stroke volume is usually around 70 milliliters. So every time the heart beats, it moves 70 milliliters of blood from the venous system to the arterial system. Heart rate is usually around 70 times a minute. Okay, so that means that cardiac output is 4,900 milliliters, which we'll round up to 5 liters. Okay, so that's what cardiac output means. It means the volume of blood that the heart moves from the arterial system to the venous system in a minute. Okay, cardiac output. So, of course, mean arterial pressure is going to depend on cardiac output, because cardiac output is the whole thing that produces this in the first place. Without cardiac output, the mean arterial pressure wouldn't be any different from the venous pressure in the first place. Okay, so it's fundamentally essential in producing this. Okay, then the other thing that's involved is what's known as total peripheral resistance, which for short is abbreviated down to TPR. So this stands for total, that's the T, peripheral, that's the P, and then resistance, that's the R. Okay, now what does total peripheral resistance refer to? This refers to these resistance vessels. This refers to how difficult is it for blood to return from the arterial system to the venous system. If it's really, really difficult for blood to return from the arterial system to the venous system, then of course mean arterial pressure is going to build up higher and higher and higher. Okay, whereas if it's easy, that's going to reduce mean arterial pressure. So these two things determine mean arterial pressure: cardiac output and total peripheral resistance. So the more difficult it is for blood to return from the arterial system to the venous system, potentially because all of the resistance vessels have constricted, let's say, okay, to all tissues of the body, then that will mean that total peripheral resistance, this number here, will be higher. Okay, right. So, coming back to why it would be dangerous to have systemic vasodilatation of blood vessels, which is what would happen if only the local effects of endocannabinoids were seen if you had systemically high endocannabinoids. what would happen is you'd vasodilate all of your resistance vessels basically, and this number would crash through the floor, and that would cause mean arterial pressure to crash through the floor. Okay, right, so that's why that's dangerous. Right, and that's why that isn't what happens when you inject systemic endocannabinoids. Okay, right, so we are now going to plot mean arterial pressure against time, and we're going to see what effect does delivering an intravenous injection of anandamide cause on mean arterial pressure, and we're going to think through why it has this effect by thinking about this equation. So that's what this entire discussion was about, trying to understand this equation. Mean arterial pressure is equal to cardiac output times total peripheral resistance. Okay, right, so basically this is what you see. So we're going to have blood pressure, or rather mean arterial blood pressure, so mean arterial pressure here on the y-axis, and we're going to have time on the x-axis. So maybe it stays at exactly this right level, 90 millimeters of mercury, okay, and then we're going to deliver our intravenous injection. Okay, so here comes our injection of anandamide. Okay, now, initially what you see is you see a little dip, okay, like so, which is called a depressor effect. Anything which causes a decrease in blood pressure is called a depressor effect, okay, so this is a depressor effect where blood pressure is dipping. And then what happens is you get a long, sustained increase of blood pressure, like so, and then this is known as the pressor effect, and this can last for several minutes, okay, so a long period of time. Okay, so this is the pressor effect of the intravenous injection of endocannabinoids. Okay, so you might be thinking, well, what on earth is this bit here? This bit makes sense from what you've told me, but this bit doesn't make sense at all. Okay, and you'd be quite right, it doesn't make sense from what I've told you, so we need more information. Okay, so this little dip actually is something very, very interesting. This is just a little side bit onto this story, okay? This basically is an example of what's called the Bezel cherish reflex. Okay, so the Bezel cherish reflex goes well beyond just the role of endocannabinoids. Okay, it's something much, much bigger, much, much wider than endocannabinoids. So let me just take a little bit of time to explain what the Bezel cherish reflex is. So basically, this is what the Bezel cherish reflex is. Basically, in your heart, so I'll just draw a little picture of the heart here. Okay, a very simple picture of the heart, so I'll just draw the four chambers. Basically, in the walls of the heart, you have pain neurons, okay? Sensory neurons that are receptive to painful stimuli within the heart, okay? And these are specifically C fibers, okay? So, basically, these are sensory neurons, and these can be activated by all sorts of horrible chemicals. Okay, so basically, horrible chemicals can activate these pain fibers here. So if horrible chemicals are in the wall of the ventricle, potentially because they have come into the blood, okay, so if you, for instance, give an intravenous injection of some horrible chemical and it goes into the chambers of the heart, then it might just get into the wall of the heart and activate a few of these C fibers. Okay, and what this then causes is these C fibers go up to the brain, okay, and carry the information up to the brain, and then the brain's response is basically a protective response for the heart. Okay, the brain's response is to hugely increase parasympathetic nervous system tone to the heart. So just like you have sympathetic nervous system tone to the heart, you also have parasympathetic nervous system tone to the heart. Okay, and in the Bezel cherish reflex, the activation of these C fibers here by some horrible chemical potentially, okay, is going to cause the activation of the parasympathetic nervous system that innervates the heart, so that PNS tone goes up hugely. Now, what's the effect of the parasympathetic nervous system on the heart? Well, principally, the principal effect of the parasympathetic nervous system on the heart is that it causes bradycardia. Okay, it slows the rate at which the heart beats. Okay, now why is it doing that? Why is that a good idea? Well, this is seen as a protective reflex. Okay, the idea is that if the heart has got these horrible chemicals present within the extracellular fluid surrounding the cardiomyocytes, then what we need to do is try and make conditions better for the heart. So one of the foremost things we want to do is make the heart cells have to work less hard, so that hopefully they can deal with these horrible surroundings basically better. Okay, the idea is that if we're forcing them to work on really hard despite these horrible conditions, the chances that they're going to die will be greater than if we drop their workload basically. So the idea is make the heart cells contract less often to try and protect them from the damage of these horrible chemicals. Okay, so what you find is that the heart rate goes down hugely. Now, what does this cause then? Well, if heart rate goes down, of course the cardiac output is going to go down. So the amount of blood that you're going to pump from the venous to the arterial side in a minute is going to go down. And that then is going to cause mean arterial pressure to go down. Okay, so mean arterial pressure, remember, was cardiac output times total peripheral resistance. So if we decrease cardiac output here, of course what's going to happen is that mean arterial pressure is going to come down. Okay, right, so basically that's why you get that initial dip in mean arterial pressure. Anandamide is going to activate C-type fibers in the wall of the myocardium and activate the Bezel cherish reflex. Now you might think, well, is anandamide classed as a horrible chemical? Well, apparently it is, because it activates certain receptors on the C fibers. So I'm going to particularly tell you about which receptor on the C fibers is activated by anandamide, and we've already seen it. Okay, it's the TRPV1 receptor. These C fibers on within the wall of the myocardium have TRPV1 receptors on their surface. Okay, here. It's a common receptor that you find on many pain neurons. Okay, so this is TRPV1 here. And basically, these TRPV1 channels are going to open, they're going to allow in cations, so sodium and calcium ions. That's going to cause depolarization, and therefore you're going to start getting action potentials firing up our C fiber, and that then is going to activate the Bezel cherish reflex. So that's how initially, when you inject in the anandamide and you give the initial bolus dose, now that's an important point actually. When you first inject it, of course, the amount of anandamide, the concentration of anandamide that will be arriving in the heart is going to be much, much higher. Okay, initially you get that bolus dose, which is because you've just injected it and it's all together at the moment, so it's not being distributed out in the bodily fluids correctly yet, and it's just in a single bolus basically. So it's the high concentration of anandamide initially in the chambers of the heart that then activates the Bezel cherish reflex initially. Then as the anandamide distributes, its concentration goes down to such a level that the Bezel cherish reflex stops. Okay, but that's what you're seeing initially, that little blip on this graph. That's caused by the Bezel cherish reflex, and it's characterized by the heart suddenly becoming bradycardic. Okay, so that's the bit that doesn't make sense then. Now let's go to the bit that does make sense from what I've told you. Okay, so we've now got a systemic dose of anandamide. Okay, so of course locally anandamide would cause blood vessels to vasodilatate, and it would cause the heart to decrease contractility. But of course systemically it's going to go up to the brain, increase the amount of sympathetic nervous system activation, and now all of the sympathetic nerves are going to start firing much more rapidly, okay, and therefore actually sympathetic tone is going to go up to higher levels than it was before, despite the fact that the cannabinoid type 1 receptors are on the axon terminals of the sympathetic nervous system neurons, and that these decrease the amount of noradrenaline released for each action potential. Basically, the rise in central sympathetic nervous system tone completely beats that, and in fact it reverses all of the effects of cannabinoid receptors and other targets for the endocannabinoids that would occur locally. And in fact, what then happens is overall the effect of a systemic injection of anandamide is that heart rate goes up, Okay, so you now get the opposite. You get tachycardia. Okay. You also get contractility of the heart going up, again because of the increased sympathetic tone to the heart, which beats out the fact that the CB1 receptors, which are actually on the cardiomyocytes, causes decreased contractility, so it overpowers this effect completely. Okay. You also get contraction of the vascular smooth muscle cells overall, and therefore vasoconstriction. Okay, so going back to my picture of the blood vessels, okay, basically sympathetic tone has now gone up so much that it beats out all of these other effects that we've just looked at, and overall causes the vascular smooth muscle cells to contract, and therefore blood vessels start constricting. Okay, and all of those effects now are going to cause a rise in blood pressure because we've increased cardiac output by increasing heart rate and contractility. Increasing heart rate means that cardiac output goes up just by that simple formula of cardiac output is equal to heart rate times stroke volume. Increasing contractility increases stroke volume. It increases the amount of blood that the heart actually ejects on each beat. Again, that's how it increases cardiac output. Vasoconstricting blood vessels then increases total peripheral resistance, and therefore that also helps to bring blood pressure up. So this is the origins of this pressor effect of giving a systemic injection of anandamide. Okay, right, so we'll call it there for this video, and in the next video what we'll discuss is one final experiment which basically allows you to get around the rise in sympathetic tone that is triggered centrally by a systemic injection of anandamide, and this is by doing this experiment in an anaesthetized animal. So a general anesthetic basically is capable of blocking this rise in sympathetic tone that's triggered centrally by endocannabinoids, and we'll look at that in the next video.